All right, well, thank you very much, and thanks for the invitation as well. I'm going to try and think about, up on stage here, some of the ways where brain control, how we learn to modify our minds, is taking new directions, and also we're learning a lot more about how to do it optimally. And I want to kind of begin just by thinking about the fact that brain modulation has been going on for a long time. People have been trying to modify their minds through the consumption of tea and alcohol and coffee or meditation. In other words, there are activities that people do that have primarily the purpose of modifying their mind, and they've been going on for many thousands of years. And so there's something, I think, built into us to desire to improve our minds and to try to recognize disorders and to also to augment everyday experience. The story of the 20th century really has been about, as you've heard, about the development of technologies to try and fix some of the neurological and psychiatric illnesses that affect us as humans. And pharmacology, as you've heard, has been one of the big stories. But there's a lot of mystery about how these drugs work. A lot of these drugs have side effects. In fact, probably every drug on the market right now for treating a brain disorder has some kind of serious side effect, at least some fraction of the patients treated. And in part, it's because there's this need to try to delve into the circuitry of the brain to figure out how they work. This is from a recent editorial about how there's sort of a general despair about the continuation of the traditional models of looking at the brain disorders and to try to treat them. Here's a quote from the director of the US National Institute of Mental Health. There's almost nothing that gives hope for a transformation in the treatment of mental illness. And there's lots of other similarly depressing thoughts that people have expressed. And it's also very hard to find drugs to treat brain disorders. So these are some numbers here where it can take almost a decade to find a drug and take it to the treatment of humans. And the failure rate is well over 90%. So this is a tremendously tough arena to be operating in, to try and find drugs that can treat brain disorders effectively without causing side effects. We've heard about electrostimulation for treating depression. This really, I think, harkens to a kind of way of thinking about the brain, which is to find targets and then to direct energy or information, perhaps through electricity, perhaps through pharmacology, to try to correct the disorders at the neural circuit level. The brain is a computer made of all of these cells called neurons, and these computations can be disrupted, and they have to be fixed. You heard a little bit about non-invasive magnetic stimulation, and the ability to use energy that can cross through into the brain from the outside is tremendously powerful. I picked this example from the lab of Rebecca Sachs at MIT because it really drives home how a knowledge of a circuit can really allow you to influence things very precisely. What she did was to do brain scanning and look for parts of the brain that are active when people make moral judgments. So, for example, you might be watching somebody named Grace, and she's making a cup of tea for her friend. And if she thinks it's sugar and then puts the sugar in the tea, most people think there's nothing wrong with that. If she thinks that it's toxic and her friend drinks the tea and dies, most people agree that's probably very bad. And so there's some cross terms here, though, that are kind of profound in how they deal with how people think about morality. Suppose that Grace thinks this powder is toxic, but turns out to be just sugar. It was mislabeled, and she puts it in the tea and her friends survive. Most people still think that's an immoral act because she has the intentionality of bumping off her poor friend. So what her group did was something very profound. They scanned the brains of people using those brain imaging techniques you heard about, and then when they found the area that lit up when people were making complex moral decisions, they then delivered a magnetic pulse to alter the activity in that part of the brain. And she could actually change the moral judgments that people could make. Suddenly, people would think that, well, she survived, right? The friend survived, no problem, And this paper, I think, was really striking for this fact that it really drives home how profound the knowledge of circuitry of the brain can be, that it has the ability to shape how we even think about ethics and judgment and morality and fairness and some of the basic things that allow us to be humans and to operate in society. And yet, as you heard at the beginning, the brain is really a computer made out of cells. And so the question is, how do we reconcile these pictures? And there's two things that I think are going to come out of this in the next really couple of decades. One is an understanding of the brain as a computer that allows us to learn something deep about how ethics and morals and emotions are implemented in the brain. And Rebecca's study that shows that perturbing a single point in the brain with a brief magnetic pulse can entirely change somebody's ethical viewpoint on the world really drives home how close to home this is. And most of us never think about this. Our minds are almost too close to ourselves. We can't observe them. It's almost a truism that it's very difficult to know what's going on in our minds. We almost always know these things after the fact. And so I think what's happening in neuroscience right now is that approaches are being taken to try and allow people to probe the brain as a circuit. Brain scanning has been very powerful at showing the regions of the brain that are involved with different normal and pathological behaviors. But what's been happening due to all sorts of different technologies, such as microscopy and the ability to look at the microstructure of the brain, is that people have realized the complexity of the wiring and the complexity of how the neurons in the brain communicate and how they change very rapidly. These neurons that are in the brain are firing off electrical pulses, perhaps some with speeds of many hundreds per second. And yet, in a tiny cubic millimeter brain tissue, you have 100,000 of these cells, each connected to 10,000 others, 1 billion connections in something the head of a pin. Now, there's some hope. One thing is that we've realized that the parts of the brain are made out of many different specific components. The cells of the brain come in many different shapes, and they're made out of many different kinds of molecule. And so if you can understand which cells embedded within this dense heterogeneous matrix of the brain are contributing to a disorder, then that can allow you to try to hone in on that cell. Maybe you can find a drug that binds to it. Maybe you can target an electrode to a region that contains these kinds of cells. And so, for example, you can see here some small star-shaped cells, which are amongst the cells that are compromised in schizophrenia. And you can also see some long spindly cells that can project long distances. And there are literally hundreds, if not thousands, of kinds of cell. The atlas of the brain still is not completely charted, as it were. But there is hope because technologies can be developed that allow you to analyze these circuits. So I'll talk about a couple of these. One of the advantages of brain research has been that it's taught us a lot about these cells. One important fact is that they compute using electricity. And electricity, as you've already heard, can be used to target specific cells in the brain. But electricity also has some disadvantages. It'll spread. And so if you want to really be able to activate a cell or to figure out what it does, or to turn it off to figure out how it might be contributing to a pathology, and if you delete that cell, maybe things get better, ideally you have some way of dealing with this very dense, heterogeneous, and messy arena. And the disorders that are prevalent affect a lot of people, over 1 billion people around the globe. And if you look at the list, many of these disorders are not even treatable, much less curable. Alzheimer's, ALS, multiple sclerosis, and many forms of blindness and pain. So the question becomes, how do you go about solving this computer? How do you develop tools that allow you to figure out which cells in the brain are involved with a problem and then to fix it, which is not obvious because you might have to pick a set of cells deep in the brain, or cells that are scattered about the brain, but around each cell are cells that are uninvolved with a disorder, and you don't want to affect them. That would cause a side effect. So really a big theme over the last several years has been that we need better tools. Technology is one of the great drivers of progress in medicine. And you've heard about technologies such as using malaria even as a technology, which seems a little bit primitive nowadays, and it's quite possible that 100 years from now, people look back at what we're doing now and say, oh, they were still giving people drugs? Can't believe it. So one of the big questions is, how do you invent tools to address these problems? And one of the tools that we've been trying to develop over the last several years, and already has started to be used widely, at least at the scientific level, has been to try to take advantage of the fact that the cells of the brain, the neurons, compute using electricity. So suppose we have one cell embedded within this deep tissue in the brain, and we want to make it controllable. Well, it computes using electricity, so what we need to do is really to install in this cell something that will convert energy into electricity, and none of its neighbors will get this converter. So one idea would be to take a solar panel, which turns light into electricity, and install it in some of the cells and not others. And then you could shine light, and that cell would be controllable, but its neighbors, which don't have the solar panels, wouldn't. And that turns out to be actually feasible. So this is a tool that is in widespread use now, where you can put an optical fiber into the brain and beam light in, and then we found molecules that convert light into electricity that can be put on certain cells and not others. And one of the reasons why this has been powerful is that we've turned to nature, which is far smarter than we are, to try and figure out how to do this. There are lots of species of plants, such as algae. Here's a single-cell algae that's swimming towards the surface of a pond. and they have to be able to sense light in order to photosynthesize. They actually have a little eye spot that can sense light and allow it to swim towards the surface of a pond. And in this eye spot are little molecules that act just like those solar cells that we talked about. When light hits them, they allow a voltage to be created. And except that instead of powering up a battery, it's actually signaling to that little tail of the algae so that it can swim. Now, of course, you've got to get this solar panel out and install it in neurons. And this took many years of thinking and not really getting anywhere. But it turns out that there's some luck, that these solar panels are completely encoded for the genome of this species of algae. And so you can take the gene that encodes for this little molecule and using the power of genomics, which has given us all of these sequences that govern the principles of how organisms work, the snippet of DNA can be transplanted into a cell, and that cell will then take up the protein, the DNA, and actually manufacture those little solar panels and install them all over the cell. And amazingly, this pretty much worked on the first try. These molecules can end up on the surface where they can then convert light into electricity. And so if you hit the brain with a pulse of light, these cells will be activated that bear these proteins that convert light into electricity, but all the other cells that are nearby, which don't bear this, will not be changeable. So this means that you can do all sorts of things now. Let's consider those cells we talked about earlier, the ones that are atrophied in schizophrenia. Maybe there's the possibility that you could take those cells, those squat star-shaped cells we talked about before, put these molecules into just those cells to make them sensitive to light. And now when you hit the circuit with a pulse of light, then you'll be able to control those cells and not their neighbors. So this is a powerful tool. You can, for example, test the theory that those cells are involved with the pathology. It could be that they're not. Those cells might have just atrophied or died as a side effect. But if you activate those atrophied cells and they could suddenly rescue some of the function, maybe you could try to figure out how the schizophrenia is occurring. So I want to talk about one example of some work that's being done by one of our collaborators, which is trying to find circuits in the brain that mediate addiction. And to do this, Christopher Fiorillo, who trained in the field of addiction, devised a very simple task that is very simple to carry out in terms of looking for parts of the brain or cells in those parts that can drive the learning of the repeating of almost useless tasks, which is one of the hallmarks of addiction. So what happens here is that a mouse, there are lots of mouse models or animal models of brain disorders, and they can serve as a proxy for the human in cases where we want to look at these detailed circuits in a way that cannot be done in the human brain, where the scanners tell us the regions, but not which tiny sub-elements within those regions are important for a given disorder. So in this case, a mouse is used, a classical laboratory animal, and if the mouse goes here, he gets a pulse of light. If the mouse goes here, he doesn't. And so if the cells that are under that optical fiber that let light into the brain can mediate addiction, that they can drive the reinforcement of whatever the mouse was doing before, then we should see the mouse go here over and over and over again. And so you can actually find cells of the brain where this happens. Every time the mouse goes here, he gets a pulse of light, and so he'll go there over and over again because he's working for the light. And so one can now try to find targets in the brain that cause these kinds of pathological behaviors. You'll see it never goes to the other one because the light is good stuff. And if you can find such cells, you can then use those cells potentially as targets for where electrodes could go, or to try to find drugs that will bind those cells and fix the physiology of those cells.